Acute myeloid leukemia and the myelodysplastic syndromes are a heterogeneous group of diseases characterized by clonal expansion of undifferentiated myeloid precursors, impaired hematopoiesis, and bone marrow failure. Under the microscopy, the cut point of 20% of bone marrow and peripheral blood blasts distinguishes MDS from AML. In the past decade, intensive sequencing efforts have dramatically improved our understanding of these diseases with the identification of different key driver mutations contributing to the pathogenesis and outcome of AML and MDS. However, most of these studies have focused on targeted DNA sequence-based approaches or have been limited to the analysis of only some specific leukemia subtypes. We know that the integrative genomic analysis, including genetic mutations, gene expression, and structural genetic changes, may reveal important insights into leukemia classification and outcome. Therefore, here we decided to perform an unbiased analysis of both AML and MDS, and here we analyze around 700 MDS and around 600 AML by integrated genomic and transcriptome analysis and by correlation with the clinical pathological features and the clinical outcome. This with the final goal to define myeloid leukemia subtypes of diagnostic, prognostic, and therapeutic relevance. This study is a part of a bigger project led by Munich Leukemia Laboratory, which has so far analyzed almost 5,000 cases from 30 different hematological entities by whole genome and transcriptome sequencing. In my presentation, I will highlight three take-home messages. The first is that we need an integrated gene expression and sequencing mutation analysis for better classified myeloid malignancies. I will highlight the importance of integrated structural variations into classification systems, and finally the importance of combinatorial gene interactions for prognosis, because we know that many genes can contribute to the pathogenesis outcome and therapeutic targeting. Overall, we identified over 7,000 variants in over 800 genes with a median of 5 mutations per case. This oncoprint provides a big overview of the genetic alterations with each patient in a column and the different colors representing different types of mutations. We found that some mutations can be shared between AML and MDS, although the frequency of these mutations may vary depending on the specific subtype. For example, we identified a higher frequency of mutations in genes such as NPM1 in AML compared to MDS, or vice versa, higher frequency of mutations in splicing genes in MDS compared to AML. NPM1 is a recurrent genetic alteration in AML, and we confirmed its frequency in around 27% of cases. But also here we showed that if we combine NPM with additional mutations, the outcome may vary. And so we identified that, for example, NPM1 and cohesion mutations can have a very good outcome compared to NPM1 and FLT3 mutations that instead have a very poor outcome. By unsupervised hierarchical clustering, we were able to define different gene expression subgroups characterized by specific mutational patterns, and we identified 9% of cases that showed the overexpression of a gene called MN1, which is a transcriptional coactivator that cooperates with the nuclear receptors for retinoic acid and vitamin D. And we showed that this group, in apparent contradiction to the good outcome of core-binding factor rearranged leukemia that's also overexpress the same gene, have a very poor outcome. So it's not only a gene, but a combination of genes that again confer a different outcome. And here we found that this group was characterized by high frequency of mutations in RUNX1. Mutations included not only sequence variants, but also structural variations, and they impaired outcome in both cohorts. This group was also characterized by TP53 mutations. They overall accounted for 12% of AML and 10% of MDS. They are associated with complex karyotype and with older age, and they definitively confer a poor outcome, even worse when they occur in patients with complex karyotype. Finally, the genomic landscape of MDS cases was really complex. So we were able, based on the combinations of different mutations and the gene expression, to define different subgroups as listed here. And again, we confirmed the importance of combinatorial mutations to determine a specific outcome. So in conclusion, the integration of mutational and expression data from such a large cohort of adult pan-myeloid leukemia cases enabled the definition of subtypes and constellations of mutations with prognostic significance. Here, some of the suggested subgroups are shown, and we will show additional novel mutations that have been identified in these groups. And thank you.